Hello students, today we are going to learn in this video about the Mendelian experiments in pea plants. So Gregor John Mendel is the first ever scientist who is experimented heredity in pea plants. His experiments in pea plants proved that the traits or characters are transferred from parents to offsprings during the time of fertilization, during the time of gamete formation and reproduction. So we can see here he experimented in pea plants with the different traits. So he first of all he selected pea plants for his experiment because there are certain distinguished characters or distinct characters in pea plants. The first thing, in their flowers, in their height, in their seeds, seed pod and color of flowers, color of seeds, color of pod, everything. There are distinct characters, different characters. So he selected. The second thing, he selected pea plants because pea plants have a very small lifespan, and within 2 or 3 months they complete their life cycle. So planting and producing seeds from pea plants is can be done within a very less time limit, within 2-3 months, and we can observe, or he can observe, the changes take place in pea plants very clearly. As it is a very small plant, he can observe manually the changes. So due to all these reasons, he selected pea plant. And after all, he was a gardener working in a convent. So, or in a monastery, not in a convent, but in a monastery he was working. So, he was growing pea plants there. While growing the pea plants, he observed it, and then he, out of his observations, he collected informations and data, and on the basis of that data, he put forward the theory, which are known as Mendelian theories. So here you can see the different colors of or distinguished characters of pea plants. Here in this slide you can see the tall and dwarf plants. See, tall and dwarf plants. Don't think that a small branch and a tall branch and a short or dwarf branch is kept here. It is not like that. The plant itself is dwarf here, and the plant in this side here you can see this plant is tall and this one is dwarf. So that is the nature of the plant. The tallness and dwarfness are the nature, or the two different or two contrasting characters of the pea plant. Same. So how some plants are tall and some are short, dwarf. How some of the characters, some of them are tall and some are dwarf. So now we can see here the Mendel's monohybrid cross. In monohybrid cross means crossing involve only one character. When crossing of pea plant involve only one character, that means one contrasting character is taken for observation. Here we can take tallness and dwarfness of the plant. That is one character, but tallness and dwarfness are contrasting. This is mentioned, the character is the height of the plant. So the character taken is the height of the plant, and tallness and dwarfness, two contrasting traits are taken here. You can see in this picture, while he was observing one tall plant and short plant, out of curiosity, he cross-pollinated the tall plant flowers with the dwarf plant flowers. So pollination, you know, the transfer of pollen grains from anther to stigma. He took the anthers from the tall plant and put in the stigma of the dwarf plant by emasculation and artificial pollination, that is cross-pollination. After that, he observed that the next generation in that plant, that flower seeds are produced, and he collected that seeds. After collecting those seeds, he planted it again. So in the next generation, he observed that the first generation itself, when he planted it, he observed that there are only tall plants. Actually, he was crossing or cross-pollinated a tall plant with a short plant, tall and short. But in the first generation, he observed all the offsprings, all the plants are tall out of the tall and dwarf parents. So why this type of all the plants are, why all the plants are tall in the first generation? Again then he thought that naturally where the dwarfness disappeared. So to find out that, he allowed them to self-pollinate. Self-pollination means no artificial methods. Let them pollinate by themselves. So allow them to pollinate by themselves. Among themselves, the plants pollinate and fertilization take place after seeds are produced, and those seeds are collected and planted again. They are the F2 generation plants. The plants out of those seeds, the tall and tall plants. Both parents are tall and both the parents are tall. Here you can see the F1 generation plants, when they are crossed together or self-pollinated together, then the next generation, F2 generation plants, you can see 3 tall plants and 1 dwarf plants arise. That means 3 tall and 1 dwarf means 1/4 of the total plants were dwarf and 3/4 were tall. So now you analyze it. The initial parents were 1 tall and 1 dwarf. The F1 generation, out of those parents, all plants were tall, and the F2 generation appeared in the ratio of 3:1. 3 tall and 1 dwarf. If there were in the F2 generation, if there are 20 plants, in the F1 generation, then what will happen? This 20 plants, out of these 20 plants, 15 will be tall and 5 dwarf. In this ratio, 3:1. So why this happens? He began to analyze it. Why this type of appearance? So he concluded, later he conducted so many experiments, and finally he concluded that the tallness is a trait, dwarfness is another trait. Both are contrasting traits of height. And this tall nature of the plant can be dominant over its short nature. That is why in the F1 generation no short plant can be seen, because tallness dominant over shortness. So the tallness is referred as the dominant character and shortness is recessive character. That means recessive character cannot be expressed in the presence of a dominant character, that is tallness. So in the next generation, how it reappeared? Because they carry some gene. If the gene for tallness is capital T and dwarfness is small t, if we cross them we can see that here you can see, I am going to do it here. If capital T is the gene for tallness and small t is the gene for shortness or dwarf nature, when they cross capital T and small t, then first generation carry genes. So there are two alleles for each gene. So we represent capital T for tallness and small t for dwarfness. It is the rule that there are two alleles, two factors are there for one character. When the formation of gamete, in the time of gamete formation, they separate out because the chromosomes, half the number of chromosomes contained in meiosis. During meiosis, half the number of chromosomes are separated out in the gamete. So the gamete contain only one T here, and here also one small t the gamete contain. So when they cross the F1 generation, plants all are capital T and small t. The plants contain gene capital T and small t. They are not pure. Here it was capital T pure for their nature of height. Small t also have a pure nature, because both the alleles are same, both the factors are same there. But here in the F1 generation you can see capital T and small t. This is a hybrid variety, means it has both the characters. It is called heterozygous nature. Heterozygous nature. Heterozygous plants are there, but in the presence of capital T, small t cannot observe or cannot show. That is why all the plants are seen as tall. But in the next generation, this is capital T, and in the next generation, when capital T and small t plants are crossed, here you can see capital T, small t and capital T, small t. इसको जीनोटाइप भी बोलते हैं, जीन का टाइप, जीनोटाइप. So capital T, small t are the genotype of F1 generation. When they self-pollinate, their gametes will be capital T, small t separated out. Here also capital T and small t separated out. When these cross together, capital T, these are two separate parents. We will do it clearly once again. Capital T and here we can do it. Here take this space, take some space and we can do it. Capital T, small t, capital T, small t. These are the two parents in the F1 generation. They cross together, so gametes are capital T and small t. Half the number of chromosomes separate out in gamete. They cross together with a capital T gamete and a small t gamete, which is another parent. So we can see here capital T will come together with capital T, so we will get capital T genotype. When capital T come together with the small t, we will get capital T, small t genotype. And here capital T small t again, capital T small t. And the last one is small t small t. Small t and small t. So one out of it is small t small t. That is the dwarf plant. Small t and small t are the dwarf plant. All others capital T, capital T, capital T small t and capital T small t. All three are heterozygous. Two are heterozygous, one is homozygous. The genes are same in one but different in two. But due to the presence of capital T, they are dominant. They are taller. They are tall. But the last one is small t, that is dwarf. So this is Mendel's monohybrid cross. So after this cross, he derived that in the time of gamete formation, the genes separate out, the chromosomes separate out, and the genes in the chromosomes separate out. And at the time of fertilization, they again come together and cross together. So, in Mendel's monohybrid cross, he proved that independent segregation of in monohybrid cross he proved that gametes are, in the time of gamete formation, the factors for characters separate out, and at the time of fertilization they join together. So this is monohybrid cross. With different contrasting characters we can do monohybrid cross. You try monohybrid cross with violet and white flowers, with violet and white flowered plant, or with yellow seeds and green seeds, or with wrinkled seeds and round seeds. For any trait you can do monohybrid cross. So try it. We will see in another video. In that we will discuss about dihybrid cross. Dihybrid cross means cross with two different traits. So till then you learn this video, hear it and take notes and learn this and draw this diagram given in this video. Okay, bye.